Assalamu alaikum to all. Today we are going to discuss about the secondary metabolites. Why secondary metabolites are produced, what are secondary metabolites, we will discuss under this topic. So, secondary metabolites, these are the metabolites which are produced by plants and are not utilized for themselves, as plants produce primary metabolites as well as the secondary metabolites. The primary metabolites are those metabolites which are utilized by the plant for its growth and development. Like if we'll talk about the synthesis of proteins, synthesis of amino acids, synthesis of lipids, these all come under the tag of the primary metabolites. But the secondary metabolites are the metabolites which are produced by plant by the metabolism or the metabolic action of the plants. So, if we will talk about under natural conditions, plants are having many potential or many enemies like bacteria is there, viruses, fungi, nematodes, insects, and even herbivorous animals are there, which are enemies for the plant. So, under natural conditions, so plants are able to develop cutin on their surface or suberin or waxes which are also there, which also act as a secondary metabolite, but these are not actual secondary metabolites, but they act as a secondary metabolites so that they will get rid from the entry of the pathogens. Like if we will talk about the cuticle, this is the waxy outer layer or the periderm, which is again the secondary protective tissue which help these plants to prevent first water loss and doesn't allow bacteria or fungi to enter into the plant cells. So this was the first primary line where the plants are not getting infected by the by the organisms, pathogenic organism. But there is again a secondary line by which these plants are able to produce secondary metabolites which help these plants to get rid from the to get rid from the to get rid from the pathogens. Like if we will see in the diagram, this is how the simply secondary metabolites are produced. The biosynthetic processes, which is utilized for the production of primary metabolites, overlaps with the overlaps with the process of the secondary metabolites. Sometimes we get confused whether this the product which is from any particular pathway is primary metabolite or the secondary metabolites. But these secondary metabolites are having specific characteristics. Like we can say that they are always produced in low quantities. Sometimes they are widespread in the whole plant or they are spread in the whole plant, or sometimes these secondary metabolites are restricted to particular organ. If we will see in the diagram here what is happening, the photosynthesis or is occurring, then from the photosynthesis sugars are forming, then from the sugars we are having another sides, from the sugars we are able to form the different cyanogenic glycosides, these are also metabolites, or then sugars when are getting involved in the respiration process, then from this respiration process amino acids are formed, then from amino acids simply alkaloids are formed, or we can say from amino acid phenols are formed, from phenols lignin is formed. This was one pathway. Then another is the from sugars when respiration is occurring, then from amino acid proteins are formed. When these amino acids are involved in the phenols, then these phenols get intermixed with the mannoline CoA, which are able to form the fatty acid lipids. From these mannoline CoA plus fatty acid lipids, they are responsible for the formation of the flavonoids. These flavonoids are the largest group of the secondary metabolites. Then from the flavonoids, there is one another compound formed that is called as the tannin. Then again, from the sugars directly saponins are formed or cardiglycides are formed. Then in another case we can see the mm, when respiration process is occurring by the respiration, acetyl CoA is formed in the mitochondria. Then from the acetyl CoA, terpenoids or steroids are formed inside the cell. This is how the secondary metabolites are formed in the plant cells. So these secondary metabolites are also the natural products of the plants. These can be used for many natural products, like we can use these secondary metabolites in case of soaps, or we are having many secondary metabolites which are having specific smells, so these can be used as essence, or these are mostly these it has been observed that these secondary metabolites are mostly involved in the preparation of the drugs. When we have many examples wherever we are getting drugs from these plants. Even nowadays, many strategies have been employed to increase the production of secondary metabolites under in vitro conditions. Like we have studied till now the bioform transformation process, where we are able to insert our gene of interest into the plant, by which this plant are able to synthesize more concentration of transgenic compounds. Or we can go for immobilization of cells. We have studied that thing also, how immobilizations are done for the production of the secondary metabolites. We can we can also increase the production of secondary metabolites by altering in the metabolic pathway, that is metabolic engineering, by altering the pathway to produce to produce or overexpress more amount of secondary metabolites by the plants. Till date, till date only the tobacco has given the positive results, in which we have taken the tobacco cells which were immersed in the cell suspension culture, and this tobacco suspension culture is mostly used for the for the production of the pharmaceutical proteins. The tobacco plant was the only plant where we have carried out many different experiments. Nowadays, we can use the cell line of soybean, tomato, or rice for the production of the pharmaceutical proteins. Till now, by the use application of the cell suspension culture, we are able to produce antibodies, interleukins, erythropoietin, human granulocyte, macrophage, colony stimulating factor, HDMCSF, and hepatitis B antigen. These are the all pharmaceutical products which are produced by in the cell suspension culture. Till now, it has been observed that we are having 85 novel compounds, out of which we are having 23 alkaloids, 30 quinones, and 11 phenol compounds. So all the 85 novel compounds are produced from 30 different plant cell culture systems. Means हम लोग 30 cell lines plant cell lines ले रहे out in these 30 cell lines we are able to produce 85 novel compounds. Earlier, when the scientists have started working on the production of the secondary metabolites, they observed that the culture condition of the plants or the cells are mainly affected by the medium which we are using for its production. So they have observed that we are having two different types of medium which are involved in the secondary metabolite production. As we know the graph curve of the plants, we are having lag phase, log, then the stationary phase. As we all know, the secondary metabolites are always produced in the stationary phase. So what is happening? The first plant start its growth when it has reached its maximum phase of lag phase, sorry, log phase. Then these plant cell enters into the stationary phase where they are able to produce the secondary metabolites. Zinc et al. In 1977 was the first scientist who observed this dual system of medium. So he worked on the production of the alkaloids from the Catharanthus rosaceous cells, where what he did, he modified he modified medium by which he was able to produce higher concentration of secondary metabolites. So zinc did, he reduced or sometimes he eliminates phytohormones from the medium. So we can say that the he zinc what he did, he reduced phytohormones or sometimes he eliminates phytohormones from the medium, and especially is the 2,4-D is the oxen. Then second he did reduction of phosphate reduction of phosphate level. When he was able to go for the production of secondary metabolites from the plant cells under in vitro conditions, there he reduced the phosphate level. First क्या हुआ, he eliminates phytohormones or he reduced the concentration of phytohormones in the medium. Second is the reduction of phosphate level and third is the increase in the sucrose level or alteration in the carbohydrate source and the nitrogen source or ratio. So this was the first scientist who worked on the dual system of medium for the production of the secondary metabolites. Then another scientist came into existence, Fujita et al. In 1981. He worked on the erythro erythrorhizome cells. He worked on the erythrorhizome cell cultures, where he observed that these erythrorhizome cells are able to grow best on LS media, Leishman's and Skoog media, 1965. But again, he observed that its production for the chiconin has been maximum in White's media, White's in 1954. So again, he observed that the two different medias are involved, where in first media we are able to have higher growth of the cell or cells are growing at the faster rate. Then in the second medium, we are having higher concentration, higher production of the secondary metabolites. This is because of the variation of the nitrogen content in the two media. Stable productions were observed when he used nitrate as a nitrogen source. When he used nitrate as a nitrogen source, he observed that there is a higher and stable production of the secondary metabolites. So Fujita devised a two-stage method system. First is for the biomass production in LS media, and then is the production media, Shikonin production media in the White's media. Then what he did, he again modified these media, which he then differently named these media. These medias were first for the growth he named this media as MG5 media, and again for the production media he named that media as a MG9 media. By altering the basal media, that is LS media and the White's media, he was able to increase chiconin production by 13 fold. इसने क्या किया, दो सिस्टम बनाए, प्रोडक्शन ग्रोथ मीडिया एंड द प्रोडक्शन मीडिया. ग्रोथ मीडिया में इसने क्या यूज किया, LS मीडिया. Then in the production media, he used the White's media for chiconin production. That is one of the secondary metabolites. He altered these both media, where he named these medias as MG5 and MG9 media, where he was able to see that there is the 13-fold increase in the chiconin production. This was how this research has started for the production of the secondary metabolites. Now, first we will go for the classification. First, we will discuss about the classification. Mainly, it has been observed that the secondary metabolites are broadly categorized into four major groups. First is the alkaloids, terpenes, phenolic compounds, and glycosides, out of which we have to only discuss alkaloids, terpenoids, steroids, and the enzymes which are formed in the industries or industrial enzymes. So first today we will discuss about the alkaloids. Alkaloids is one of the major groups which include 20 12,000 different 12,000 different alkaloid species or different compounds has been observed. These alkaloids are all the alkaloids are having three basic characters. First, all the alkaloids which has been isolated till now are able to be soluble in water. First, these alkaloids should have at least one nitrogen atom. And the third, they exhibit high biological activity. When there is at least one nitrogen atom in their compound, so they are able to accept a proton. If they are able to accept a proton, that will give a slightly basic nature to the compound, so alkaline, hence they call this as a alkaloid. This alkaloid is a heterocyclic compound, and although a few aliphatic non-cyclic nitrogen are there, like we are having different examples, it has been observed that the total alkaloid means 12,000 alkaloids are there, 20% of which are produced by the flowering plants. मीन जो हमारे अल्कोलॉइड्स है कितने 12,000, उसका जो 20% हिस्सा है वो कहाँ से आ जाता है, फ्लावरिंग प्लांट से. एंड मोस्टली दीज आर herbaceous dicots. These are mostly the herbaceous dicots. The word alkaloid is a virtual synonym of word with the drug, but it has been observed out of 12, 10 are utilized as a drug. It has been observed that if we are able to isolate the 12 alkaloids, out of this 12, 10 are used as a drug purpose. So these alkaloids are able to respond in human. These alkaloids interfere with the neurotransmitters. If we will accumulate or if we will consume higher concentration of alkaloids, these are that time toxic. Or again, if we will use lower concentration or smaller doses or smaller quantity of these alkaloids in day-to-day life, then these are act as a therapeutic. Like we can say that these alkaloids are used as muscle relaxants, tranquilizers, painkillers, poisons, or even sometimes these alkaloids act as a mind altering drugs. These act as a mild altering drugs. As we all know, one of the oldest alkaloid is opium. Yes, it's Opium. This opium is the immature seed capsule. Yes, opium poppy. The scientific name of this opium is the Papaver somniferum. In 2500 BC, there were a tribe called as Sumerians. These Sumerians used to mix this opium with wine whenever the person is in pain. That time this opium along with the wine are able to induce sleep. When the person is in bad condition, when they are having the pain, acute pain, that time what they used to do, these Sumerian people used to mix opium with the wine in 2500 BC, and this mixing has induced the sleep in that time. Since then, these secondary metabolites are known to the different humans. If we will talk about what is opium, it's a latex gum which contains 20 different types of alkaloids. It doesn't have single alkaloid present in it, but it has 20 different alkaloids present in it. So we are having different alkaloids, like we are having here morphine, codeine, and pipervidine. These are the three different alkaloids which are present in the opium. So in all the three cases, we can see this is the structure of the codeine. This is one of the secondary metabolites which is present in the opium. This is the morphine. As we can see, at least one nitrogen atom is present there. And the another class is heroin. We will talk about this also. So all the three compounds are having at least one nitrogen atom. All the Three, like morphine, codeine. Opium is a semi-synthetic one. It's not naturally occurring compound. So all the three, opium is mostly in, like, it's present in Mediterranean area, like Iran is there, Afghanistan, Pakistan, even some Southeast Asia are able to produce this opium. If we talk about the general classification of these alkaloids, they are having a ring system present. They are having a ring system present, and the heroin, which is there, is the semi-synthetic alkaloid, which is mostly produced by the acetylation of the morphine. Morphine and codeine are the two naturally occurring the alkaloids. So when we go for acetylation reaction to the morphine, that time the heroin is produced. We are the basic precursors or simple precursors from which these alkaloids are formed are few amino acids: tyrosine, tryptophan, ornithine, arginine, and lysine. Again, I will repeat, we are having few precursors from which these alkaloids are formed. The common amino acids are tyrosine, tryptophan, ornithine, arginine, and lysine. As we know, another example is there the tobacco. Tobacco is able to produce nicotine, and we all know it is synthesized from by the nicotinic acid. And caffeine is the pure derivative. Caffeine is the pure derivative. These are the naturally occurring alkaloids which are present in the nature. Even we are having some alkaloids which are specifically produced in specific organs, like these alkaloids may be produced in the, like these alkaloids will be produced in the, like roots, leaves, or young fruits. Like we are having one example of in matlab, like these individual alkaloids, these are called as individual alkaloids. Why? They are specific to the specific organs. One of the best example is berberine. This is present in one of the berries. This is mostly seen, this type of alkaloid. So this berberine is mostly seen in the seeds of barberry plant. The another class of this is the lupinine, lupinine, lupinine. This is the secondary metabolite or the alkaloid produced by the genus Lupinus. This is toxic to those animals which are grazing, like grazing cattle, sheep are there. So these are poisonous to those animals which are grazing cattle. So mm, when these lupinine is consumed more and more, that time death occurs. We are having another datura is there, which is also having able to produce the secondary metabolites. Even we are having two different solanaceous plant which are able to produce secondary metabolites. First is the potato, another is the tomato. If the alkaloids from the potato and the tomato is consumed more, that time death is occurring. But naturally, by the wish of Allah, these potato and tomato are not able to produce that much of secondary metabolites which will be harmful to us. So they produce very less amount of secondary metabolites. The secondary metabolites produced by these are alpha salonine. These are produced by alpha salonine, and the secondary metabolite another is the tomatine and alpha salonine are the two secondary metabolites which are produced by the potato, Solanum tuberosum, and the tomato. So these both, if taken in higher concentration, that time death will occur. But mm, god has made its God had made these, or we can say that God has made such a these secondary metabolites are made in such a way that they are produced in very minute quantity. These are not toxic to us, or they are synthesized below toxic level. We are having another class of this secondary metabolite alkaloids is indole alkaloids. As you know, it may be, as name indicates, These are having indole basic ring, which has been derived from the tryptophan. It has been derived from the tryptophan. These second alkaloids are, like we said, these secondary alkaloids are muscle relaxant. So these secondary metabolites like alkaloids are having antioxidant property. So secondary metabolites act as a scavenger for free radicals, or they can be also used as a metal chelating agent. Even they have also ability for hydrogen donation ability. These secondary metabolites are also antimicrobial in nature. De Luca was the one scientist who worked in 2006. De Luca worked in 2006 found that the imidazole derivatives having an immense therapeutic potential and also reported their antimicrobial activity. So another class is there, the alkaloids class, which are mostly and at the top of like for their therapeutic purpose. These two are most important. This is vinblastine and vincysteine, vincristine, vinblastine and vincristine. These are the two secondary terpenide indole alkaloids which were used for many cancers, which were used for treatment of many cancers like Hodgkin's lymphoma, leukemia. But these are important for therapeutic purpose of the cancer, but their concentration is 0.05%. So before applying or before giving these secondary metabolites to plants, we have to go for downstream processing, plus we have to go for purification and extraction of these secondary metabolites from the Catharanthus rosaceous plant. So while proceeding for the second or downstream processing, these are very costly. So it's better not to use these products because they cost more than $1 billion for its purification. Even as we said, we are having another class, that is the cocaine. These secondary metabolites are taken from the leaves of cocoa plant, or we are having codeine. These are extracted from the opium plant, or we can say we are having nicotine. These are taken from the tobacco, or caffeine. Simply caffeine is taken from the coffee beans or the leaves, and all the together they are widely used as a sedatives. So this was about the alkaloids.